This is the Relationship and Biodiversity State Lab. You need to know genetics and ecology for this lab, which consists of 7 tests total. This video demonstrates tests 4 and 5. The goal of this video is to find which of the mystery plant species is closely related to Botana curis through a series of molecular tests, paper chromatography and Enzyme M test. Paper chromatography is an analytical chemistry technique used to separate the colored chemicals or substances of a mixture. For this technique to work, there is a special filter paper that is used. As you see here, we have our filter paper with a line at the bottom and pigment identification labeled at the top. BC is for Botana curis, and then species X, Y, and Z. Using a clean microtip dropper for each plant extract, place 1 drop of each pigment at the line, at their corresponding location. Then place the filter paper in a cup of water. As the solvent rises on the paper, it meets the sample of mixtures and carries the components of mixtures along with it. Along the way, the various components of the mixtures are separated out at different levels on the paper. The components of each mixture can separate based on solubility, molecular weight, and interactions between paper and the mixture. Let the paper stand in the cup and remove it before the water reaches the labels at the top. We are left with a chromatogram, which is then analyzed based on how the pigments separated. Record your observations of the colors and relative amount of pigments in Table 1. Here are the observations. Botana curis, we see pink pigment first and a bright blue pigment at the very top. Species X, we see a faint pink mixed with a darker pigment and then a blue color at the top. Species Y, we only see blue pigments throughout. Species Z, we see pink pigment and then a bright blue pigment at top. Note, the pink and the blue pigments for Botana curis and species Z appear at the same level on the chromatogram. Our next molecular test is for the presence of Enzyme M. Since it's not practical to test the plant directly for curol, we can test it for Enzyme M. If the enzyme M is present, then the plant may produce curol. Remember that in a chemical reaction, 2 substances come together and form a completely new substance. In our reactions, we saw that when we mix a solid enzyme indicator with a liquid plant extract, some of the mixtures created a new substance, gas, which is evident by the presence of bubbles. If there is no bubbling or fizzing, then no reaction took place. For this test, put a small scoop of the indicator powder into the depressions of the well trays. Then use clean microtip droppers to add each plant extract to the wells and observe the reactions. Here are the observations. Botana curis fizzes a lot and produces a darker blue color. Species X does not fizz and has a darker color. Species Y fizzes a little and produces a lighter blue color. Species Z fizzes a lot and has a dark color. Now record these test results in Table 1. Note, if we see fizzing, then it is a positive test for Enzyme M. Once you have recorded these in your data table, continue with the other molecular evidence tests to finish this lab.